few years ago, I read an article about microbes in plants and soils. And I've often wondered while I'm digging in my garden if I have the right plants, right microbes for my plants and soil. You know, is it like good cholesterol? You want certain ones and not other ones. And of course, there's a lot of effort in looking at microbes because there's hundreds and hundreds of them. Yeah, so I thought, well, I wanted to learn more about this. So I decided to look around and see if I could find a real scientist that did published in peer-reviewed literature and had a pretty good reputation for his work. And lo and behold, by golly, there's one right in our backyard at U of H Clear Lake. So that was pretty easy. Dr. LaMontagne, I hope I'm saying that right, got his MS degree in microbiology from the University of Massachusetts, and then went on and got a PhD in biology from Boston University. As part of his thesis work, he did research on the nitrogen cycle in the marine sediment. We can sort of relate to marine sediment around here, and that the ability of the bacteria to remove the excess nitrogen, as all of us know, there's runoff from agriculture as well as from our lawn fertilizer that pollutes our bays and streams. So he became interested in manipulating the properties in such a way that it would help the plants and help the water quality. So tonight he's going to be speaking on bioprospecting, the plant microbiome to improve the yield and sustainability of agriculture. Michael, you want to take it over?
Yes, thank you. As Helen said, I'm at the University of Houston Clear Lake. I would be right upstairs if we're in the typical room we meet in. I'm also the founder and chief science officer for Endobiome, which is a bioprospecting company, microbial discovery company that I co-founded with a colleague at the University of North Texas Health Science Center. Now, I have some slides here that are just for introduction, and I'm leaving them in here for the benefit if anyone wants to look at this slide deck. I'm not going to read the text to you. I'm going to jump down to the big picture. So the big picture is to prevent widespread starvation in the developed world. We're not talking about the developing world, but here and in Europe, we need to double the crop yield per acre in the next 30 years. So this slide is from a paper published in 2013, and the dots show the trends in yield, which is on the y-axis, per acre for the major grains upon which society depends. Now, the solid lines are just fit to this mob, to the dots, and extrapolating out to where we will be in 2050. So we will have this much soybeans, this much wheat, etc. The dashed lines are where we need to be to meet the calorie demands of our growing population. That is, we will be increasing yields, but there's a gap that will lead to a crisis of an existential crisis for our society. Now, how do we close that gap? Well, heretofore, the main strategy has been dumping a lot of fertilizers and pesticides on the fields, as well as manipulating the variety of the plants. So we have GMO crops, we have tons of fertilizer, and a whole bunch of pesticides. The amount of fertilizer we're adding is already quite high. It's hard to see that we can go much beyond where we are now. Just to illustrate this, this is a picture of a plot from a trial we did in Arkansas when I worked at a company in North Texas. And that plot was the grower's recommended amount of fertilizer per acre, 260 pounds of nitrogen per acre, which is on the scale of this guy here. And if we multiply that by the hundreds of millions of acres in cultivation, you can see that we're already adding a lot of fertilizer. And that fertilizer may be changing the plant microbiome with unintended consequences. To address this, we yanked up the plants at harvest, pulled out the root ball, and did microbial community analysis using next-gen sequencing techniques to define the microbial community that's living in these roots. These microbial communities have formed close associations with the plant roots, and presumably they're there for a reason, either to help the plant or to take nutrients or away from the plant, but in some way interacting with the plant. We define that as the plant microbiome. Now, this figure here on the y-axis shows the results of that bacterial community analysis. The metric diversity is a combination of the number of species as well as the evenness. The y-axis is a metric of bacterial diversity. It's not just the number of species. If you count, 5 trees in a field, then you have a diversity richness of 5. But if there are just one of those trees is a fir tree, and then all the others are deciduous trees, you wouldn't say it's a very even distribution of the plant community. This index incorporates both the number of species as well as the evenness. The x-axis is the amount of nitrogen assimilated by the plants as measured by leaf nitrogen. We need to have the plants assimilate nitrogen so that they can grow. And we need to increase that growth, obviously. Now the symbols correspond to the different treatments, where we have no fertilizer treatments down to urea fertilized treatments. And what we could see was a negative trend where more nitrogen in the plants corresponded to less diversity. Now, the question is, does that matter? And we could hypothesize based upon ecological theory that it does. As a society looking at native plants, I don't have to tell you the importance of having diverse plant communities for the health of the system. That hypothesis was really first proved by Tillman, who published a paper where they manipulated the diversity of plants in plots in prairies in the upper Midwest. And what they found is if the plant plot had more diversity, more grass species, then the field yielded more plant biomass. And this can be measured just as true diversity as number of species, diversity of plant types, like C3 grasses, C4 grasses, etc. And subsequently, not only the yield has been shown to increase with diversity of plants, but the resilience of the community. Now, the dogma for microbes was, and to some still is, well, so what? The theory that was first postulated in the 1920s by Bergernik, and which can be summarized as everything is everywhere, but the environment selects. So what this holds is that there are so many bacteria, that there are trillions of bacteria in soil, millions per gram, diversity measured in thousands of species, that it couldn't possibly matter if you added one, took one away, or did any manipulation of the microbial diversity. And that theory was, again, the dogma when I did my thesis, and my thesis advisor was not quite, looked at me like this when I proposed to go look at the microbial diversity in sediments. The alternative hypothesis is that the microbiome matters. And we hear this all the time as it relates to human health. I'll give you an example, not of men, but of mice. In this paper, they looked at the relationship between the microbiome of mice and obesity. What they did in the in the treatment group is they exposed the mice to penicillin at a very early age, and that permanently altered that treatment of antibiotic permanently altered their microbiome, as shown by different colors of bacterial symbols here relative to the control. And those mice that had been exposed to antibiotics when they were pups grew up to be fat adult mice, as shown by this cartoon. Interestingly, at least to me, is when they transferred the microbiome from the fat mice to a germ-free mouse, they ended up with fat mice. And when they transferred the microbiome from a skinny mouse to a germ-free mouse, they ended up with skinny mice. Where the germ-free mice were mice raised with no significant bacteria, raised in a plastic bubble, essentially. So we can reject the hypothesis that the microbiome doesn't matter, but we don't really know how it relates to plant growth. I can't at this time tell you that these are good bacteria, these are bad bacteria, and that you need to have these particular bacteria for your soil. We do know that more bacteria is generally better, and more microbes is generally better, as illustrated in this figure, these two figures. So on the x-axis, we have fungal abundance and bacterial abundance, respectively. On the y-axis, we have yield of peas. It's actually pea hay per acre. And the symbols correspond to the treatments, whether they were conventional fertilizers or organic fertilizers. And there was a positive trend between the amount of bacteria and yield. And it would follow then from species energy and area theory that more equals more diverse, but the data is still out on this. That is, we don't have a strong link between the diversity of soil and productivity. There's one recent paper that tested that hypothesis by looking at potato yield in Quebec. And the hypothesis is, as I stated, that more diverse soils would be more productive. They collected potato soil from 13 farms in Quebec and measured bacterial diversity using the metagenomic sequencing methods that we used in Arkansas. Basically, take the soil, grind it up, take the DNA and sequence it, and then use that to infer the number of species. The different metrics include the one that I showed before, which is the Shannon Diversity Index. And what this plot is a little busy, so let me walk you through it. This is a one-to-one plot of yield versus yield. So that's self-evident. But if we jump down here and look at yield versus diversity, this number here being a negative number suggests that diversity goes up, yield went down for those plots overall. And it depends upon the metric you use, but none of these relationships were particularly strong. So the authors came up with an index called the species bacterial index for potatoes that suggests that these particular species, and identified which particular bacterial species were beneficial for those particular fields. And not based upon that, but ahead of that, there have been microbial products developed for agriculture, and some of those are widely used. Most of the microbial products developed for agriculture are members of the Bacillus genera. Of course, you've heard of Bacillus anthracis or anthrax. These are actually closely related to that species, but their niches, they occupy the plant roots and appear to have evolved a beneficial relationship with the plant to make bigger plants, which attracts herbivores to them, and then they can get in the zebra and infect them. But these are widely used for a variety of microbial products for agriculture, including biopesticides. The largest microbial product by far is BTI, which was discovered in Israel, and it makes a crystal that is lethal to insects and is a highly effective, therefore, as a biopesticide. But they're also highly effective bacillus species that are biofertilizers, as well as biofungicides. All of them have the advantage of being spore formers, so you can make a product that has practically infinite shelf life, that are easy for the growers to apply to the fields. And you can literally go and buy these biofertilizers off the shelf in farmer supply stores. As I mentioned, there are a lot of microbial products out there. Most of those are the microbial pesticides that I've talked about, but there are microbial soil treatments, and the market in 2012 for these was measured in the billions of dollars. Now, if you want to penetrate this market, which we do, not just for economic reasons, but for the ecological reasons, that is, if we can make the plant microbiome more efficient, we can get more yields per acre with less fertilizers, less pesticides. And the trick appears to be, well, threefold. One, can you grow them? Can you grow microbes in the soil that heretofore were not cultured? Would you know them if you found them? If you grew them on a petri dish, would you know that they were beneficial and worth investigating further? Or are they something that someone saw before? And could you use them if you did? If you found a great microbe, could you get it out to hundreds of thousands of acres in Iowa? And all of those challenges are well established, but the dogma is that you can't grow them. At least that was the dogma 20 years ago when this table was made. This table summarized for different environments, including soil, the estimate percent that could be cultured if you took the sample and spread it on a petri dish and then counted colonies. The number is was at that time less than 1%, excuse me. Now, some systems showed relatively higher percentages, and this appears to be related to polluted water systems where there's a lot of whatever you want to call it in it, like activated sludge. Selects for microbes that are relatively easy to grow in the lab. But this dogma has been disrupted through the development of advanced cultivation techniques and the advent of high throughput microbial identification methods. The company I founded is based upon our approach, which uses a system we call the GRIPS, gnotobiotic, rhizobacteria, isolation, plant system. Where gnotobiotic is like the germ-free mice. These are plants that we've surface sterilized at the seed level and grown in a sealed system so that they have no natural microbiome. We can then inoculate them with a slurry from, let's say, some native plants that someone gave me, or right now students are working with plants from salt marshes to find microbes that help plants tolerate salt, and grow them under controlled conditions where the only microbes in this system are the microbes that we added to them. In this system, then the plant serves as the petri dish. The plant provides all of the nutrients and signal molecules that we cannot possibly formulate in the laboratory. And by iterative processing, where we grow plants, take the microbes on them, reintroduce them, we can select for microbes that are beneficial to the plants, identify them by next-gen sequencing. We take a slurry from the plant roots, grind it up, sequence it, and then use that sequence information to design better media and ultimately isolate bacteria that were not previously available. And we have a patent protecting this method, and it has yielded microbes that perform well in greenhouse trials. at least relative to the industry standard. So this isolate here, EN821, is shown with the open circles. The x-axis is the concentration of microbes added to the soil in terms of spores. This is a bacillus species. And the treatment effect corresponds to the size of the plant increase overall, from where 0.1 is a 10% bigger plant. And what we found is that our microbes did it at least as well as the microbes that are now being used widely in the Midwest for plant growth promotion. So we can isolate bacteria that were previously not isolated, but a major improvement has been the throughput of this discovery process. And here we are dependent on, much like we are for much of this microbiome research, on the human microbiome. So over the last few decades, 2 decades, there has been a fundamental shift in the way we look at microbiomes. That is, we discovered that we could sequence the microbiome, and that led to identification of novel species in various systems like the human gut. So the number of species identified went up linearly over time. But if you notice, the number of new species discovered shot up about 10 years ago. And that is the groups, there's really one group that is responsible for that, and they coined the term culturomics, which if you're that productive, I guess you can make up the word yourself. And what they have shown is that by using lots of different cultivation techniques and isolating thousands of bacteria, you can find new bacteria that were never found before. And the enabling technology is a mass spectrometry method called MALDI-TOF. What this technique involves is you spot the bacteria on a metal target, shoot it with a laser, and it creates a cloud of peptides. The peptides are accelerated towards a detector through an electric field, and the little ones get there sooner than the big ones. And that gives you a pattern that is a fingerprint for that microbe. And if you have a matching pattern in your database, you can say, aha, I've seen that before. If it's different, then you're like a new criminal in the FBI database. We now know who you are because we've seen that fingerprint before. And for bioprospecting, the key thing is you want to look through lots of them until you find something new that's worthwhile pursuing further. The benefit of this technique is that you can do hundreds per hour for pennies an isolate, while the conventional method of DNA sequencing still costs, let's say, $20 an isolate if you're doing just a ribosomal sequencing, and hundreds of dollars an isolate for doing the whole genome. And this group in France really accelerated their work over the last 5 years. And their most recent paper, they isolated almost 1 million colonies. From those, they generated almost 3 million mass spectra and identified 1,000 new bacterial species. And I'm not going to walk through this whole figure here. I just wanted to jump to where they are now. Where we are now is they have identified more than half of the microbes in the human microbiome. Not identified, excuse me, isolated. So for that system, we're no longer walking blind. We have representatives of the microbes that are carrying out all the functions in the system. And our goal here at UHCL and at Endobiome is to apply that approach to the plant microbiome. The trick is we don't have the resources to do millions of microbes, and we don't have the proprietary software that enables microbial identification. Indeed, when we're looking at the plant microbiome, because it's so poorly studied, most of them don't have any match in the database. 2/3 of them, indeed, in the study I'm going to talk about next, are not represented in that in the commercially available databases. I'm showing here what the spectra looks like from one of those runs. These are two standards. They're called bacterial test standard. And these are the peptide peaks that you would see if you blasted that standard with a laser. The little ones on the x-axis in terms of the mass get there sooner. And each of the peak corresponds to a particular peptide. For me, all I care about is the pattern, and I try to match the pattern. As you can see, if you ran two of them, they're very reproducible. That is, you can't tell that there are two spectra run here because they're so consistent in their pattern. And what we focus on is identifying the peaks that are reproducible between the spectra, because sometimes you have noise and then you have a signal. And what is the appropriate signal to noise ratio is what we worked out with the maize microbiome. This is a collaboration with UH Downtown and students working in several microbiology courses I teach at UHCL. What we did is we took the rhizobacteria and the endophytes from two varieties of maize, the heritage variety, a heritage variety, and the hybrid variety, where the hypothesis was that the heritage variety might support a more diverse microbiome than the hybrid variety, which is used in row agriculture. We wrote custom scripts to sort those isolates based upon their mass spectra into distinct or coherent MTUs, which is what I call the Multitoph taxonomic unit. We don't know what they are, but we know that that pattern is a match to another pattern, and we have several of them that are closely related. Then we use that machine learning, wrote other scripts to implement machine learning to identify microbes every time we saw that same microbe. And this figure shows how the similarity between two spectra is associated by Multitoph on the x-axis compares the similarity for the gold standard of bacteria identification, which is DNA sequencing. And what we found is a sigmoidal relationship where if you got above there's a cutoff where you can have bacteria that are 100% identical to each other at the DNA level for this one particular gene, but are quite different on the x-axis in terms of the similarity in terms of Multitoph. The take-home message is that Multitoph, which costs pennies for an hour, using these scripts was more sensitive at distinguishing closely related strains than the current standard that is used for bacterial identification. And with this approach, we were able to compare the diversity associated with the heritage and the sweet variety, which is the hybrid used in row agriculture. This Venn diagram is showing MTUs that are shared between the two microbiomes and MTUs specific to the two microbiomes. The genera are indicated here when they're known. For some of them, we still don't know what the genera is. We have to do follow-up sequencing projects for them. And the numbers indicate how many of each of those particular microbes we identified. Now, this is not an impressive number of microbes compared to the millions that are isolated from the human microbiome, but it suggests that we have a method for rapidly and inexpensively identifying microbes from the plant microbiome. Now, the question is, what are we going to do with them? As I said, having a great microbe doesn't do any good unless you can get it out to the field. And I bring this more into the talk as a future direction. This title of the slide is from a paper written in 2006, where they basically said that growing pots in a greenhouse is more of an art than a science. Potted plants in greenhouse are subject to artifacts that make the results very difficult to reproduce. For example, if you overwater the soil, you'll generate zones of hypoxia, which would change the microbial community and also have negative effects on the plants. There are dramatic light and temperature fluctuations in the greenhouses, particularly in a place like Texas, when in the summer it's almost impossible to keep the light at a reasonable level and the temperature at a reasonable level for reproducible results. By reproducible results, I mean you'd want to get the same results for your trial here as they do in Minnesota and vice versa. And even under controlled conditions, lab-to-lab variation is quite high. But just to illustrate what I'm talking about, the variation is, here is, in this paper, they compared the variation in temperature between night and day in the field versus a greenhouse. And that, in this case, for the air in the room, the temperature was well modeled by in the greenhouse. But for the soil, the fluctuation was much greater for the greenhouse. Now, there's another system that we use, which is called a growth chamber, which is more controlled than the greenhouse. Basically, it's a box with lights and air circulation to maintain growth conditions. And you think that would address these concerns, but that does not appear to be the case. This is an influential paper from France where they grew 3 varieties of a model plant in 9 different labs. So here are the 3 varieties. Colombia, I don't know what this one is and this one, but this is a widely used variety of this Arabidopsis plant. And they sent them to 9 labs and looked at various growth parameters like leaf area. All of the labs were given the same protocol, typed up. You can see it in the paper. Add this much soil, water it this much, do it exactly this way, under controlled conditions of light and humidity in growth chambers. But as you can see, the variation in growth between labs, lab 1 had twice the growth of lab 4, and we don't know what is going on in lab 5, but there's dramatic differences in growth between labs that exceeds any differences between plant varieties, let alone microbial treatments. So we're at the stage where we've had to rethink what it takes to grow a plant. One strategy is the new field of plant phenomics. And I know everyone is sticking omics in front of it, but this one is actually backed by a lot of money. The plant phenotyping facilities use robots and conveyor belts to replace the human factor in plant growth experiments, basically to cut out the green thumb. And these systems rotate the plants daily, so none of the plants are exposed to hot or cold spots in the growth facility or high light and low light areas with any particular bias. And they're measured daily for plant growth, as well as soil moisture and other parameters that are related, that obviously control plant growth. Problem with these facilities is they cost, well, this one at A&M costs $3 million, and they cost probably that much a year to run. So it's not an option for us. What we're doing is growth chambers with an array of relatively small plots where we can grow wheat and maize and model plants. This is our collaborator at UMass Amherst who's growing Brachypodium with a high degree of reproduction, which will hopefully give us reproducible results. But I don't have any good data to show from that system yet. So in short, we think that plant growth promoting rhizobacteria can help society avoid the coming food crisis. That is, they're a tool that we can use to maintain and increase productivity. To get there, we need advanced cultivation techniques, which we have some indication, can recover beneficial bacteria from the plant microbiome. We also have to apply high throughput tools like MALDI-TOF to this microbial discovery, but the plant growth trials will remain a bottleneck for the foreseeable future. And I'd like to acknowledge the support for this. Since students were doing this work in lab, we count their lab fees as part of the funding for this. I received support from the National Science Foundation while writing the paper on the MALDI-TOF technique. Phi Tran was an undergraduate doing this work and received support, and Tori Fungate Mullins helped as part of her lab project in environmental microbiology, a course that I teach, to start this project. And I'll entertain any questions.
Well, this is Helen, and we have one question which I thought was pretty good. And you can see it when you look in chat. She says the farmers might add the bacteria or the right kinds of bacteria you can buy commercially, but then they go ahead and use inorganic fertilizers. You know, I guess the question is, isn't that a conflict?
No, it's a very good question. We're not going to get there without inorganic fertilizer for the foreseeable future. The company I worked with in Dallas actually sold their microbial fertilizers as a coating on fertilizer pellets, because the data that we had from greenhouse trials as well as field trials suggested that the microbial products could help the plant utilize applied fertilizer more efficiently. So what we're striving for is a 20% reduction in fertilizer while maintaining the same yield. So the microbes can act apparently to increase applied fertilizer use efficiency, or nitrogen use efficiency in particular. They can do this for a number of mechanisms. One mechanism that we look at is they can increase the abundance of the amount of lateral and fine roots, which allow the plants to obviously capture more of that fertilizer. They can increase germination, because what you want to do is capture the fertilizer when it's applied in the spring. And to do that, you have to have the plants grow quickly, fast, so they can get that fertilizer before it seeps into the groundwater. And that all depends upon the timing as well. Is you also want to capture get the plants in the Midwest to grow fast early, because that will help them capture the moisture associated with spring rains. So I guess what I'm trying to say is the microbial products have to be integrated into the current growers' protocols for those fertilizers, for them to be used efficiently or used at all. And that's actually a tremendous barrier to entry in the field.
Thank you. We have two more questions here, pretty good technical questions. We have an 18-foot deep lake that is dammed up. We recently dredged it, and I want to use the silt that was at the bottom for my garden. Should I use it?
I think you would want to send your soil to a soil testing lab, which can measure parameters as far as silt, clay, sand in your soil relatively inexpensively. I think it's about $10 to have your soil analyzed. And they could tell you if your soil would benefit from any particular amendment. And you could send them the dredge material as well. I would dry it out first. Or if you had a admixture in mind, like you're going to mix 10% into your garden soil, you could create that mixture and send it to them and see what they have to say. So they'll give you back a reading that would say, for that mixture of silt and your soil, how much, you're not going to give up, how much is a good amount to add, or if adding it improved the soil quality, just in terms of how they assess soil.
We have another question here. Where I'm applying these concepts is coating Austrian winter peas and partridge pea seeds with bacteria for planting the seeds. My understanding is that this is needed to add nitrogen to soil. Is this aligned with what you're seeing? And are there any general thoughts about this?
Now, is this for production or, well, I guess it doesn't matter too much, but I don't know that it's needed to add nitrogen to the soil. I think well, it sounds like this is a commercial product of rhizobacteria, which would be a nitrogen fixing bacteria that in general, it is found that adding nitrogen fixing bacteria to the soil each year accelerates the formation of nodules, and you have more nodules per plant and more nitrogen fixation if you do that treatment. So does it work? Yes. Overall, it has been shown to work even for fields which previously were inoculated, where there was previously grown that crop with that inoculum. So adding it again next year may help too. Another way of saying that is the microbes that you add as a seed coating don't appear to stick around until next year, or at least not enough to create a significant nodulation. Will it spread to adjacent? I don't know. Adjacent plants, they're not going to the microbes can't move from row to row, for example. I don't even know if rhizobacteria, rhizobium, excuse me, which is the species generally used as a biofertilizer for legume crops, is modal. I don't think it is, but even if it was highly modal, for something on a micron scale, traveling a centimeter is for us like, walking to Dallas. I mean, I don't have a good example of it, but they have a very hard time moving around. They can be carried by dust and advected with water flow, but as far as reaching another row of your fields, not likely. In general, they actually test for these growth response by split row trials, where they'll run the tractor down and treat 3 furrows with 1 microbial or fertilizer treatment, and then whatever furrows they get for the next treatment would be whatever the width of that tractor run, and they would have split rows down across the field. So the assumption there is that there's very little edge effects and that treatments are kept to individual rows.
We have another question. Have there been studies looking at the use of microbial products in vineyards to improve grape production and quality?
Well, the quality is the issue. There of course is an interest in the microbiome of all fruits and vegetables, but in vineyards, there's actually a fair degree of, well, the interest goes up with the value of the crop in terms of investing in expensive treatments because you anticipate a significant return of investment. So high value crops. Now the flavor profile of wine depends upon flavonoids that are produced by the plant. And those flavonoids are, what tells us is the difference between coffee and sugar. I mean, everything that makes up a plant that we like about them depends upon these molecules that they produce. Now, these secondary metabolites are not produced to make for our benefit. They're part of the plant's defense system. They either are there to deter herbivores, and that includes deterring microbial, not microbial pests, but insects and other herbivores. What we found by accident in one greenhouse trial is that plants treated with microbial products were much more resistant to an invasion of midges, or I can't remember, I think it was aphids, which happened across the whole greenhouse, than controls. And the way that we believe this works is that the exposure to a little bit of microbes and their particular molecular patterns tricks the plant into thinking that it's infected. And that initiates a part of the so-called plant immune response, which in a way is a parallel to the way our innate immune system works. So that the plant starts producing these defense compounds at a higher level when exposed to these microbial products. Just like when you are exposed to the co-stimulatory molecules in a vaccine, you have a reaction of tenderness at the site of infection, swelling, maybe a fever. All of those are your immune response reacting to co-stimulants in the vaccine. The plant doing that actually makes it more hardy and resistant to other pathogens or herbivores that are attacking it. At least that's what we postulate. And we showed in growth chamber experiments that this manifested in terms of increases in expression of genes for flavonoid production in the model plant of Arabidopsis that I mentioned before. Now that was work that I did at a company and that I mentioned, well, I didn't mention my name, but they're up north of Dallas, and they received a patent for that particular application, increasing flavonoid content by microbial treatments. I don't know how it's, what the status of that science is now.
We have another question. This really relates to the kind of work some of our folks are doing. If you transplant a prairie plant into a garden with a good bit of soil around the roots, are you transplanting its microbiome, and will it spread to adjacent plants? I think you've already answered that part of the question. The spread to adjacent plants are the same species that were grown from seed.
Yeah, I mean, I think you would transplant its microbiome. Whether that's permanently going to change the microbiome of your soil is an open question. Certainly, I think it'd be healthier for the plant to bring some of its microbiome with it, so it could establish itself in the new system. But at the same time, we don't think that added microbes stick around long. And this is something that investors and reviewers of proposals have brought up. How are we going to sell microbes if the microbes establish themselves in the soil? Why would the grower pay for something that more than once? And what we've shown is that, well, not something I've shown is that yes, microbes will be detectable in the soil for a couple of years later, but the microbiome does not appear to be permanently altered like it is in the human microbiome with manipulation.
Well, we use a lot of mulch in our gardens, and does that change the microbiome?
Yes, I think there's a slide on that. So, organic fertilizers, I wasn't involved in this paper, but presumably what they're talking about here are compost products. And we have one in this tree paper here where we have fresh poultry litter, which is another organic
Oh, yeah. Yeah.
product.
Yeah.
And these change the microbiome. I haven't shown that so-called beta diversity analysis where you're looking at the change in species, not the number of species, but it clearly does influence the microbiome in terms of more diversity relative to conventional fertilizers. And I want to point out that these organic fertilizers here were applied at the same mass of nitrogen per acre as the synthetic fertilizers. So there does appear to be a change in the microbial community associated with, let's just call them organic fertilizers. And the more organic matter you add, the more microbial abundance you're going to have in the soil, which, as they showed, at least for these systems,
Okay.
more microbes meant bigger plants.
Yeah. Another question, in an undisturbed prairie, will a given species of grass be found to have the same bacteria and fungi co-evolved in its root system?
I got to ponder this one a little bit.
Well, the person asking is really smart, so
Okay.
just to intimidate you more.
Undisturbed prairie. Okay, so we managed to find one somewhere.
There are a few in the world.
As in, right. So in a natural undisturbed grass, if we're comparing the microbiome of the plants there to the microbiome in a disturbed system, we would expect to have differences in the microbial community, even though the plant are the same. I would say yes. And if it was an undisturbed system, that would be probably the ideal place to find microbes that have evolved a close relationship with that particular plant species. Because we would think that as the question suggests, that the plants and the microbes would have co-evolved in that particular system for thousands of years, if not hundreds of thousands of years. And that would be an ideal place to go bioprospecting. Now, I think I saw Cindy on this talk before. If I was looking to test this hypothesis, I don't know if she's still here, I would ask Cindy to look at undisturbed marshes, salt marshes, versus the restored marshes that are being planted in Galveston Bay and similar systems. So that you have marshes that have developed, plants that have developed in those marshes since the last ice age, and there's been very little activity, direct activity as far as, tractors, et cetera, in those systems. And I believe that the microbial communities in those would be quite different in the natural marshes than the disturbed marshes. So what can you tell us? Are there different microbes?
Well, Cindy's still on, and she says, I'm still on, and we're doing just that in a project with Lee College. So great minds think alike.
And for the human microbiome, there's some funny stories about this. For example, some people have asked for and received money to get the microbiome from Amazonian tribes, pre-industrial tribes. I'm trying to of hunter-gatherers in the Amazon, on the presumption that their microbiome is more natural and more highly closely evolved with the human host than ours. And my company did a project for a company that makes a microbial product for skin called Mother's Dirt. And this skin microbiome product seeks to replace the part of the human microbiome that we lose every day we wash with soap. And the founder of that company redeveloped his microbiome to more of the natural form by not taking a shower for 20 years. And then isolated those bacteria to make us a skin product. And we provided isolates from the marine environment for that system as well, because your skin is actually quite salty. And we hypothesized that microbes from the ocean would help replenish your skin microbiome. In particular, your skin microbiome has bacteria that convert the nitrogen in your urea in your sweat back to NO, which is a vascular dilator. It helps your blood vessels expand and blood to flow. It is indeed the active ingredient of Viagra and similar products. So this company actually protects the restoration of sexual function through the application of skin microbes from a guy who never showers. And all the applications of it. It was quite an entertaining patent to read.
Oh my goodness.
And if anyone has a greenhouse and wants to do trials, we're looking for greenhouse facilities in the Houston, Galveston area that we want someone with a green thumb for this work. It's actually quite hard to find someone that can do a greenhouse trial for the reasons that I outlined before. It's not that easy to grow plants, and certainly not easy to grow plants reproducibly and generate valid scientific results. Thanks for taking the time.